we will talk about Soratase-Mediated ligation, which is considered a gift from gram-positive bacteria to protein engineering. Protein engineering is a process by which novel proteins with desired properties are developed. This field is very important as it plays a central role in biological science, biomedical research, and biotechnology. There are many applications for protein engineering, and we here discuss a new enzymatic protein ligation tool as a new method for protein engineering. The general descriptions of Soratase enzyme. Soratases are transpeptidase enzymes found in most gram-positive bacteria. They are positioned at the plasma membrane and are responsible for covalently anchoring a variety of surface protein to the cell wall envelope. Among the several isoforms, the Staphylococcus Soratase A is the focus here. Soratase enzyme performs its action by transpeptidation mechanism. Soratase A first recognizes a sequence of leucine, proline, X, threonine, and glycine within the polypeptide 1 and cleaves the amide bond between threonine and glycine with an active site cysteine 184, generating a covalent acyl enzyme intermediate. The thioester intermediate is then attacked by an amino group of the oligoglycine in polypeptide 2, which allows the ligation of the two polypeptides by a native peptide bond. What are applications of Soratase-mediated ligation? First, the introduction of new functionalities into proteins. One of the most straightforward and important applications of Soratase-mediated ligation is a method for introducing unnatural functionalities into proteins. With this new technique, various molecules such as chemical probes, synthetic peptides, and polymers have been incorporated into the LBXTG tagged recombinant proteins. And these are examples of unnatural compounds that can be incorporated into recombinant proteins within this approach. Second, the chemoenzymatic synthesis of neoglycoconjugates. This method provides a new way for introducing glycans into peptides and proteins. It is an important approach to obtain homogeneous glycoconjugated polypeptides by linking pre-synthesized glycosylated building blocks to other peptide segments. Here, figure A represents Soratase catalyzed peptide sugar ligation to obtain neoglycoconjugates. Figure B represents example of oligopeptides, amino sugars, and aminoglycoside antibiotics that can be used in this study. Finally, the covalent immobilization of proteins onto solid supports. Many protein modification strategies have been adopted to achieve the site-specific covalent immobilization of proteins. Soratase-mediated ligation also become an attractive alternative approach by providing the ability to attach protein onto solid supports in their native state, which is essential to produce a variety of technological tools such as supported catalysts, protein sensor chips, and protein microarrays. This figure here represents Soratase-mediated ligation-based immobilization of proteins onto solid supports. All information in this presentation were obtained from this link. This presentation was prepared by Hadir Hussein, Tuka Tahir, Iman Muhammad, Nama Ali, Hiba Magdi. Thanks.